The overall goal of this protocol is to perform a high-throughput forward chemical genetic screen on Arabidopsis seedlings.
With this method, we can efficiently and effectively discover novel small molecules that affect plant physiology. These discoveries will provide the foundation for further studies into physiological processes, such as cell wall synthesis. The main advantage of this technique is that tens of thousands of small molecules can be screened in a short period of time.
Though this protocol is designed for the model plant species, Arabidopsis, it can easily be adapted to other organisms, such as insects, microorganisms, or cell culture.
We first had the idea for this protocol when we learned that the university's Genomics Center needed to find a home for a surplus liquid handling robot. We immediately volunteered to take it off their hands.
Prepare the dilution library and the multi-channel tip-wash automated labware positioner, or ALP, as described in the text protocol. Manually load the stacker 10s attached to the stacker carousel. In hotels A through D, first load one box of AP96P20 pipette tips in room 1. Then, load 4 96-well V bottom plates in rooms 2 through 5, with 2 upper plates containing stock concentrations from the ordered library and the other 2 lower plates empty. Additionally, load one box of AP96P20 pipette tips in room 6. Load 4 96-well V bottom plates in rooms 7 through 9, with the 2 upper plates containing stock concentrations of the ordered library and the 2 lower plates empty. Set up the deck with 300 milliliters of water in the reservoir on P3 at 300 milliliter 70% ethanol.